Hi, everyone. Um,
But
First of all, I'd just like to acknowledge the Dharawal people, the traditional custodians of the land that Pip and I are coming to you from at Mount Annan today. Welcome you all to the first, I think it is the first Myrtle Rust Practitioner series for 2025. It did take us a little while to get things together, but we're here and we're really happy to welcome Brandan Espe from James Cook University, supervisor Environment and Grounds, I believe, to discuss with us the establishment of their Myrtle Rust susceptible collections, how that was done, and some of the challenges that he faces in continuing with that work. Um, Thank you very much. Thanks, Brandon.
yep, I'll jump on and try and keep it quick for questions. So yeah, I'm the supervisor of environment and grounds for James Cook University, and I manage what's called the TropEco program, which is the sustainability and environmental management platform for the university. So yep, and across the top we've got some pretty myrtles. So the presentation summary, the first part was to start about obviously the risk we all know, but in particular some selected components that I'll talk about further down in more depth, ex situ conservation alternative benefits to conservation, and prior requirements in particular as well that we use to kickstart. Prior requirements are probably not super useful to smaller organizations, but certainly for some of our larger ones, it's a big win. And then, yeah, phases 1 to 4, we broke them into as our primary phases. You can do as many as you want to. And then the challenges and what the future looks like for our program. So moving across the top is a quick run through of some images. We've got Backhausia tetaptra on the far left, which is the cuttings. That's a critically endangered species. And that species was actually critically endangered before Myrtle Rust even got to it. So it was a really big concern. There's less than 200 of them. It was instantly selected as a high priority for us. So for the university, our first step was to go local. And this species, and then next to it's a translocation site that some volunteers are working in, was the first cab off the rank, which was kind of sold as a restoration project, not a full-blown conservation project. And I think that was an important stepping stone because our management team was a little bit scared of jumping into full-blown conservation initially. Next to that's Rhodamna arenaria. This is part of a recent project we've moved into in Cape York, and it's a very reclusive species in the Cape that's found in a handful of fractured rainforest and dry rainforest and vine thicket environments. So we've currently got a project working with that over the next 2 years. And then we've got Eugenia rhynewoodiata, with a bit of a contentious one as to whether that form's native to Australia or not. But nonetheless, the species has a strong cultural relevance to traditional owners in North Queensland and Central Queensland, as well as through Papua New Guinea and through the Pacific Islands. So it's an important species that we sold as a cultural component as well, not just conservation. So the risk we all know, I'm assuming if you're at this seminar, you know what Myrtle Rust is, so we'll glaze over that. We've got 4 examples here, and I'll expand on these a bit later on. But initially, what I wanted to touch on was that they've got 4 very different sort of backgrounds of myrtle rust. We've got Rustantia gouldii, which was our already vulnerable listed species, and based on our surveys, we think there's less than 200 of these left as well. It's only restricted to 2 known mountains, or there's a third population that we think is legitimate. We need to check at high elevation, that's sort of like 600 to 800 meter elevation mark, 1 random population for 1,000 meters, and explicitly in riparian zone. So highly isolated, really hard to get to. It takes a whole day to hike into. Lloyd can vouch, very hard to find. We did find them in the end, but they're severely impacted by myrtle rust, and so isolation was the big factor that drove us towards the selection of this species. Restantia pachisperma, on the other hand, is very different. It's really easy to get to. It hangs over a lot of roads. It's found in some parklands. It's or was a fairly common borderline pioneer species in the lowland areas. Grows to a massive tree larger than this stockwellia behind me even. And it was a least concern prolific species until myrtle rust came around, and has now jumped to critically endangered because of dramatic population declines, poor recruitment, poor fruiting, and has these huge, large lettuce-like leaves you can see that are really highly susceptible and capture a lot of pustules. The next one we've got Lindsayamyrtus racemoides is a different one again. So it's a really important ecosystem engineer like Tristaniopsis. It's sort of common along your waterways. just like the Canuka, and it forms a monoculture in many areas. So if you lose this species, you actually have mass degradation of these banks, which then sends sediment down the creeks and rivers into the Great Barrier Reef. So it's now a Great Barrier Reef problem as well, which is another selling point we use. But this species is a little bit annoying because unlike many of the other species, there are so many populations with zero myrtle rust and then other populations being decimated. So it's really challenging to work with. And Syzygium glenum was selected for our program quite highly because there's less than 24 individuals known, 16 that we've actually come across, and it was already listed as endangered because of that before being severely impacted by myrtle rust with no known wild recruitment that we're aware of in the last decade. So ex situ conservations, the alternative benefits you can get out of this. Living laboratories for the university and for botanic gardens is obviously a big one. Having these sort of gardens that can be used for teaching and conservation and research and ongoing public engagement that have multifaceted approaches as opposed to a single approach. A really great example of that outside of the myrtle world that was predecessor to JCU is obviously the Raising Rarity program that the Royal Botanic Gardens have set up. And obviously that program focused around rare and showy species, whereas ours is focused around some sort of dreary trees in some cases. But they have also some aesthetic individuals, but more importantly, really, really important ecosystem services that was important to capture, and that's what makes them great for teaching. So exciting experience for students. Many of these are threatened species and are disappearing from the wild and are hard to get to. So from a teaching perspective as a university, it was a cool point to sell that we could show students specimen that they otherwise would never see in the wild. Cost reduction for research is another really big one. Some of these species we've had to helicopter into. Some of them we've had to go hiking for days on end. As you may know, if you've done field work, that can be quite expensive. And so having these specimens correctly catalogued in collections makes them infinitely cheaper to work on. And we've had some specimen collected for genetic analyses from the campus as opposed to the wild, because the wild populations would have taken days to access, whereas for the campus, it's a case of a researcher attending campus, grabbing a coffee, walking outside and having their samples before they finish their coffee. So it's a fantastic system for collecting, way better than field work in some cases. Safeguards at-risk flora is obviously a really big one in the media as well. PR is a big thing for funding. So being able to show that you're protecting threatened species is an easy sell to the public. And then also feeds into great public relations where a lot of people come on campus, not just university staff and students, but members of the public to see our gardens, like many of the botanic gardens here, I'd imagine, to see what's out there. And it's a really cool way to tell stories to these individuals. And then you also get the excuse to work with like-minded partners, which is sometimes challenging for us being so isolated. But it's always great to work with, state and local government, other botanic garden organisations, especially the Royals in particular, which are better developed than we are, and to steal things from them. And to build those collaborative partnerships and learn from other areas that we may not have expertise in, or vice versa. So image-wise, we've got Rhodomyrsa fusa, the velvet myrtle. This is a severely impacted pioneer species that pretty much explicitly lives where rust is most active. It's really its target environment, these sort of just exposed high wind shear rainforest environments in sort of wet swallows. Obviously, the Raising Rarity program we spoke about earlier. Collaborative-wise, we've got some staff from the Australian Tropical Herbarium, the Royal Melbourne and Cranbourne, I believe, attended that trip, and the Australian National Botanic Gardens as well. So that was a cool trip chasing not just Myrtaceae, but other threatened species as well, as a kind of extension to Tromps. And then we've got some student interns carrying out a translocation project, planting out the vulnerable Eucalyptus paedoglauca. So prepared assets in kind certainly make it a lot easier to attain funding. Being able to prove, one, that you've done these kind of projects before also helps, but being able to show that you have the resources to do these projects without having to build it from the ground up was certainly a major assistance for us, which a lot of universities and large botanic gardens and council groups certainly have the ability to draw on. Obviously, smaller NGOs not so much, but I'd recommend that they partner with other organisations like some of our local groups have. For our case, we have a well-established living laboratory already, so this is more of an extension. Each project we bring in, our threatened species project, our cycad project, our myrtle project, are more extensions of a greater program. We've got the interns and students already keen, so free labor is always useful, and they're keen to get out in the field whenever they can. So everyone's always super excited to go on these trips and learn what they can and get experience in the field. And if you take the time to train a handful up in depth in particular, they're phenomenal assets, but you need to take that time to train them initially, or they're just not going to be great assets for the program. And especially design nursery, where in the image in the bottom left, you can see pots with individual drippers to them. So we can have arid land species next to rainforest species, which allows a broad diversity for us in that hardening area. Looking at the other images, we have a student with the geode on their head. They've finished mapping the tree that they're mapping, and they're now just writing notes so the geode doesn't drown. That's a highly accurate mapping device. And we've got some students going for a swim in a creek in the Daintree, well outside of crocodile territory there, up in the hills. On a trip, we were chasing some rhodamnia and rhodamyrtis. So phase one of the project was to obviously develop and sell the program. You can't just kick these things off the ground. The first one was to write a paper to our higher ups. The next step of that was harassing them a lot and complaining to them a lot and having tantrums. The next step is then giving in. And so you have to spend a lot of personal time on that side to get them at that point, or at least I did, to get the program off and running. And then once you get buy-in from there and you've got a budget and you can sort of kick off, start small. I'd recommend requesting a smaller program. Don't go big. You're more likely to get it, and then expand that program. And that was what we found we had to do, as opposed to jumping into a phenomenal size project, which it's now sort of grown into. Somewhere between, you have to make sure you have all the permits and licensing to actually complete this in place. In Queensland, if you're collecting from private property, for example, and it's a least concern of vulnerable species, you actually only need permission and from the landholder, and you can act under Schedule 40 of the Act. Whereas if you look at endangered and critically endangered, you then need to start looking at state permits, or if you want to go any state forest or national park, again, instantly state permits. So it's a bit of a confusing thing. I think it's about the same statewide from what I can gather, but sourcing those correct permits is the first big step you want to take, because in many cases it can take months to attain them. So there is a slow process to that. Another one is then obviously going for external funding if you're unable to or do not attain enough internal funding, where there's a lot of grants that come in around conservation and wellness. Funnily enough, we actually also pulled some funding out of the Gambling Association towards well-being, and so part of the trail that went in was actually for fitness and health and outdoors experience, and happened to have these things planted along it as part of the project. So thinking outside of the box for not just conservation funding is another great way to go as well. And then obviously you have to recognize that your seniors came up with this idea all along and it's their project. That's the most important part to get continued support. So once you've kind of got to that point, you can then continue sort of capturing the benefits of the program and how it goes on further. So in our case, we were talking about how these species, such as that Lindsay and Tristaniopsis of Canucas, are excessively important in ecosystems as retainer systems and engineers, how things like Melaleuca in particular form sort of monocultural forests almost. So again, really important species, important food source. So cassowaries and tree kangaroos were a good selling point for us. They love myrtaceous species, thank God. But also things like the corran lorikeet that everyone loves in this photo, eating a gossip in Willie Eye, is a really good sell because everybody knows these colourful birds. So it's an easy thing for the layman that may not be across some of these more exotic, interesting things that you're trying to sell that you may think are cool, but if they don't come from an environmental background, they don't know what you're talking about. So it's really good to be sort of grounded with that. Important economic and traditional use values are really important as well. Obviously, we've seen funding historically go to agriculture, in particular around like backhouses. Historiadora initially had some, being an economically viable source, but also traditional values are really important to work with as well. And some traditional groups are really keen to jump on board if you have a key species in their area, and it makes the program go so much smoother. Taking that into account as well also is important to capture that history of being an oral group that's potentially going to be lost if it's not retained. Flow on values for the Great Barrier Reef is another really big one for us in Queensland in particular. Obviously, it's a World Heritage. It's a phenomenal site. It pulls in a lot of tourism. So there's a lot of industry around it. There's fisheries around it. So there's large scale funding based around that asset, the reef itself to Australia. So kind of tailing onto that, saying, oh, dirt will get into the creek if the trees erode, and really simplifying that, really, and specifying that, that will then increase turbidity and cause issues in the mangroves, will cause issues in the reef, and having that sort of story and connecting that has also been useful. So, and another point is obviously aesthetics. And I don't mean the pretty creeks and streams in the images. I mean that a lot of the Myrtaceae, such as Lithomertus and Calatrix, are stunning species in landscaping. So phase two, this is the boring part. This is the desktop work and deep questions, unless you're one of those people that like sitting at their desk. So the first thing to ask yourself is, what do you want to cover? Do you want to cover, in our case, Townsville? Do we want to do Cairns as well? We have a campus there. Do we want to do North Queensland, Northern Australia, the tropics? Selecting that area you want to cover and work in is a really important key, and then try and stick to that if you can. Don't try and branch out too much or you'll overextend. Another one is which species are likely to be impacted or known to be impacted. So in our case, we were lucky that we had a separate project for threatened species that captured species that were impacted that weren't known to be. So we got that information quite early. But certainly working with species that are well known to be impacted is another easier way to, one, add value to the collection and program, but also add likelihood to get funding. Which species are feasible for your site is another great one. We would love to grow things from the highlands, for example, here on the Townsville campus, but we're somewhat limited to essentially species below 1,000 meters elevation. And even at that 800 to 1,000 meter elevation, it's touch and go for us. So realistically, we didn't want to capture highland environments like the Tromps project did, which is why those plants went down to Sydney and Melbourne, because they were better suited to those climates than they were to our climates. What are the project costs going to be? You need to anticipate field work, salaries. Is there going to be in-kind commitments to that? All things to take into account. Do you need a nursery built? Can you maintain a nursery? Is that space rented? All of these costs should be accounted for before you start looking for funding, because you need to know where you're coming from before you start approaching people about the program. And then you can sort of stop. And at this point, you'll have a headache. So this is where you do your daydreaming and think about lovely places like the bottom of my screen at Bishop's Peak. And then you get back to real work and you look at how will you accession and collect your data, what data are you going to be collecting. That's a really important component. If you get halfway through your project and then decide you need new data, essentially the first half of your project is now void because you're missing data components. So it's no longer really the same collection. And that can be a big issue. So really knowing what you want to get out of the program and what information you need at the start is a key component to starting any of these programs. Otherwise, you may not collect elevation, for example, and then you may find out elevation is an important factor later on in the project, and you've got to go recollect it from everything if you didn't collect it initially. So that's really important to do. And then how you're going to do the materials. So with Myrtaceae in the wet tropics, certainly seed production, if they're impacted by rust, is significantly lowered, if at all existent. And so cuttings are the next step. And for us, we find that semi and hardwood cuttings are the most successful, whereas I understand in temperate climates, softwood cuttings are a more commonly used approach, and certainly for herbaceous things. So it would depend on, again, how you're going to approach these species and how you're going to collect them and how do you intend to. Air layering is another potential option if they're in a highly humid environment, but it often doesn't work if they're highly susceptible to rust and being impacted severely. And then obviously at extreme cases, excavation. But if you start looking at that side of things, good luck with permits. How you grow these on is another really important one to consider. So this tree behind me is a Stockwellia. These are one of the largest trees in Australia. It's not something you want to put near a footpath or near a building. It's going to destroy it. So being such a large specimen, you need to take into account the size, and you don't want to be planting, 50 trees a metre apart, only to have to cut every second one out in a few years' time. So it's important to consider that as well. And you certainly don't want to plant things on top of utilities that are then going to have to be removed later on because they've broken a $100,000 high voltage cable. So really important to consider that one. How will you be establishing these and how are you managing them? So you can put something in a parkland and that's fantastic, but if that parkland's planned for being developed into housing in 10 years' time, then it kind of defeats the purpose of putting a conservation practice program in. So where are you going to put these things and who's going to maintain them to establish them? Because you don't just set and forget. It's not concrete. You have to look after them for a few months at least to establish them, even if you have a set garden, as I'm sure everyone here would know. And then how are you going to ensure that data is retained long term? So what's going to happen when you leave the project, or what's going to happen if you jump onto another program? Is that data stored and properly in place somewhere and is accessible to multiple users in case you do leave the organization. So, and then obviously finally, again, back to permits, really can't stress that enough. That's really something we see hold a lot of researchers up when they're geared up for a program. They get funding and then they have to wait 3 months for a permit. Phase 3 is field work. So this is the exciting part. Lots and lots and lots and lots of safety performers that I'm ashamed to say I palm off to other people. So activating your field working, hoping it's not going to be torrential rain, which up here is a major one for us. Obviously, you want things that are in active growth. If you're doing cuttings or you want to know, are they fruiting or flowering? What time of year are you going to be going out there? What kind of data are you collecting? As an absolute bare minimum, if you're going to do a conservation program, I would recommend collecting obviously the name, a code which is unique to the individual. That code can be whatever you want. To keep it simple, we've just used a genus and species and then a clone number, and the lat, long, and elevation. Additional things you might try to capture is susceptibility of the rust damage, what kind of soil it is it basalt, rhyolite, what kind of slope are you on, what kind of regional ecosystem are you in, what kind of microhabitat are you in. So a really good example is back out to Taptra that's found in eucalyptus woodlands, but if you actually go and collect them, they're only found in dry rainforest and small rocky outcrops within those woodlands, which will dramatically change how you're going to maintain them in a collection. So that kind of data as the field worker is going to be really useful to the horticultural side of the program and establishing them. But also, if you're looking at translocations, you need to know the companion species, the pollinators, the vectors, the substrate, the water availability, underground water availability. All of these factors come into a major role if you're going to go further down the road of translocations as opposed to just a garden collection. So that's an important one to capture. Phase 4, which is the final phase for where we're doing with our one, is obviously establishing the collection. This is where we're up to now. So selecting the right site for the right plant. Like I said earlier, you don't want to put a giant tree right next to a building or on top of utilities or near water, like a piping. And you don't want to put a lowland shrub from a rainforest out in the paddock in full sun. So taking consideration on where you're going to be placing these is a really important component for long-term success of the specimen. Ensuring the right care is already in place. So is the site irrigated? Is there sufficient composition for the soil? You can see that we've got a Rhodamnia sessiliflora in a garden on the top right page, really thick leaf litter, really rich organic soil, but quite airy soil so it doesn't stagnate and hold water, which the species thrives in. And that tree, I believe that photo was 3 years ago. That tree's now about 6 meters tall, and we probably get about a kilo of fruit every few months dropping off of it. So it's performed fantastic, but that's because it's in the right location, not just because we're cheating and being in the tropics. And ensuring assessing and mapping is carried out as you go. So you can see a tree planting plot on the bottom corner. Every single one of those had a metal tag placed on them and was mapped and added into our database as they were going in, because you don't want to plant them out, have a pile of tags in your hand and be sitting there scratching your head going, which was which. So it's really important that as you're going and not as a, I'll deal with it later or you will lose the data. So translocation versus gardens is another one. Are you looking at doing a large scale translocation like you can see on the bottom right for this eucalyptus or of your Frabachausia? Or are you looking at collecting for a garden? If you're doing a translocation, you need significantly more individuals for a higher germplasm representation, whereas if you're doing it for a garden, you can get away with less, but obviously the more germplasm representation and genetic variability, the better the collection will be and the more value it will have. So, and educational signage is another big one to draw people in and continue attaining funding for it because it increases interaction, which is a really big thing in attaining funding. At the bottom, we can see my leading hand, Hayden, installing special irrigation for this taxodium, which is actually a pine. But either side of him is also, or in front of him, outside in the garden, a Tristaniopsis that were installed into this water feature, the deep cell garden with a heavy watering regime to specially suit these specimen of full sun, which they get in their riparian zone, but also a high water retention within that bed. So it's essentially a swamp garden in full sun. So again, that correct location, and these trees are now 3 times the size as they were just 2 years ago and are already flowering and fruiting. But again, the right location was the most important part of that, and that ongoing care and establishment to get them where they were. And above, we have a master's student holding a, I believe, Epinendrum, the native one. in our nursery with a whole bunch of plants behind her that she was involved in caring for and planting out and really quite intensively containing the program. Challenges, the big ones we faced at least, was initially our team wasn't skilled in this environment. They were more designed for sort of generic landscape care and sort of mowing in that. So we've had to upskill the team, and a couple members of the team have taken an interest in identification and getting involved in these programs to the point that when we did position reviewing PDPs at the start of each year for what their jobs were for that year, two of our staff as special projects actually requested to be part of these programs and install special gardens on sites, and we're really excited to be involved. So not all people do jump into that, but there are some staff that really moved into it, and so we've sort of helped push them along that way. Pathogen outbreak and monitoring. So canary in a mine is a term we use. In Townsville, we're really lucky. We're quite a dry, horrible climate, so we don't really get rust for the most part, unlike our Cairns campus where we've avoided our Myrtle program. But nonetheless, we actually, to make it easier, selected a species, Eugenia ryan and a population from Cape Tribulation that was one of, if not the most susceptible population of myrtles we'd ever seen for rust. It's actually now locally extinct, that population. And we put them through our gardens every 100 to 150 meters. And that's what we check for myrtle rust, as opposed to the whole garden. We can just check one specimen, and we've now checked the whole garden. So it's significantly quicker. And we tried to put those near footpaths and doorways where people are more prone to brush against them and touch them and come in contact with them to increase the chance of them getting rust should it arrive to campus. Underground utilities and destructive trees are another really big one. So there's a syzygium just on the top right behind me, this giant stockwellia. We've got Restantia powering up through the center photo on the left. These are really large trees, and essentially as far as the canopy reaches is where you're destroying utilities. So these things needed to be 20, 30 meters away from any utility, with the exception of irrigation. Water and so much water. If you're doing rainforest species in particular, it sounded like a great idea for us to take them to a dry climate because they wouldn't get rust, but we have had to water them so extensively. And in later times, we actually then started developing more organic rich components and using inline fertigation systems and wetting agents to cut down on the water usage, but still allow us to grow those plants in our drier climate. There's less rust prevalent. So yeah, wetting agents and inline fertigation systems and automating those systems was a major step to maintaining the collection long term that we didn't consider initially. The size of mature trees, not only for utilities, but also, looking at this building on the bottom left, you can see some trees growing up through the center of the building. These are quite small species. If you got excited and were to put a Syzygium glenum or a Stockwellia or a Syzygium ericalyx in there, you wouldn't have a building by the end of it. So really selecting where these things are going to go and managing that. And on top of that, anything larger, and your solar panels are now null and void as well in this case. So there's a lot of other factors that go into it, not just you have a tube stock to put in the ground. You need to consider what will happen in 20 years' time when that tube stock is now a 30-meter tree. Wildlife interactions was another really big one for us. So wallabies in particular and scrub turkeys are the bane of our existence here. They are forever digging things up. They are forever eating things. So initially, some species do need to be guarded from them. Some species do need to be cared from them long term. And usually once things are established, it's not an issue, but initially wildlife is a big issue for us, and keeping them out of areas until it's established enough that they can harass it is a key component of what my officers do. Future planning and development, again, you don't want to pick a nice empty field, plant it out, and then find out in 10 years' time it's residential housing. It kind of defeats the purpose. So for us, we have a 50-year master plan. It's under re-review, but the programs have been incorporated into that 50-year strategy. So we have a longevity around these collections and where we're putting them. That's a really important one that I've seen a lot of our local groups, certainly in Townsville with our land care, make the mistake where they've selected sites, put a lot of effort into it, and then it's being bulldozed and turned into a road, which really defeats it, and it's quite disheartening. So looking into that planning and looking into that future core plate for that is really important to consider and to take on. And then the next step is obviously, and final step is beyond. What are we doing, and what would you likely do with these collections? So specimen sharing, as we were just chatting about at the start with us sending stuff down to Dermot and Lloyd and that at Royal Melbourne is a really important component of conservation, and having meta collections and backup populations and having more than just one person holding them, because if we were to lose them, then you've lost all of them. So it's important to share these across the board and have a meta state. Selective breeding potentially in the future is another one that we're not looking at this point, bar the back house to Taptra, where we're going to be more intensively breeding as opposed to selective. But it's certainly something that's worth considering for threatened species across the board. Seed banking where you're able to. Obviously, there are many unorthodox myrtles out there. A lot of our rainforest species just do not like being stored. And that's where ex situ conservation of physically growing these plants becomes really important. And so I feel that those species are more so inclined to be the targets for ex situ as opposed to species you can seed bank readily. Cryopreservation, obviously, the next step beyond that. Teaching and learning, again, do you want to use your collection for preaching the problem and getting it out there and expanding it to more people? And obviously, the more people that know about it, the more likely there's going to be action. Susceptibility testing is another one in controlled conditions in particular. It's really hard, and I'm sure most of us have come across this in the field where you'll go and find a population like the Lindsay myrtus where they're being decimated by rust, and then you're like, this is ruined, and you'll come back 6 months later and there's absolutely no rust. And that's not a really uncommon occurrence I'm finding talking to other practitioners in particular. So susceptibility testing under the same conditions is a really good way to gauge how susceptible are these actual species, broad range. And then also taking into account what conditions do they live in general and trying to replicate those conditions as that general board of their susceptibility. And then obviously ongoing monitoring. These kind of programs aren't a chuck in the ground and walk away as much as we'd love that. There are ongoing programs where if you've gone to the effort of going out and collecting a threatened species from some unforgiven hellhole of a mountain, you don't want it to die in a couple of years' time because you've moved on to a new project. So although we're focusing a lot on other species at this moment outside of myrtles, we still have a heavy focus around the myrtle scene as well. And we're still not only getting new species in, but we're constantly monitoring and assessing our existing species and making sure they're still going well and monitoring their health and tracking how we need to monitor and care for them moving forward. Not just sort of going, yep, that flavor of the month is over, move on to the next one, move on to the next one, move on to the next one. You really need to focus on what you've built and create that strong foundation to ensure the growth of the program and success of the program. I think that's everything I've got for everyone.
Thanks, Brandan. Have we got some questions for Brandan? Either pop them in the chat or raise your hand on the screen. I have one. Lloyd, and then Veronica's got one as well.
Hey, that was great. Thank you, Brandan. Great as always. Just on that, we were talking before about the preference for cuttings material, and maybe Dermot, you can get involved in this as well. But yeah, I did notice that when we were up doing fieldwork together last year, that you guys were taking a lot more hardwood cuttings. Do you think that in future, when we're collaborating in the field, that it would make sense to do hardwood cuttings for us as well for some of these myrts? Like, is that more of a response to your climate, or it's more of a response to the particular trees themselves?
I think in this case, my background from what I've been taught was really focused with hardwood, so that was really my personal preference. But we have found some species where I will only do semi or I will only do tip cuttings, and there is certainly a special preference where if I'm working with a syzygium, for example, I'll almost never do tip cuttings because we've just had such success with tropical syzygium, whereas hardwood and semi-hardwood have been so successful. And rhodamia and rhodamyrtus hardwood, we're getting almost 100% rates. It's insane. And then we move to semi and it goes down a bit, and we move to tip and it goes down dramatically. Obviously, seed is a big preference because you can get a broad range of individuals, like a single seed will have more than one individual. But if you're not able to collect seed, there's an issue. And the other side to that is you don't know who the other parents are. Whereas if you want to look at selective breeding in the future, I think it's important, if you're not going to be including genetics, to know exactly which ones you've got from where, because the parent may be from kilometers away. And so we personally prefer cuttings for that clinality tracking.
And I suppose if you're sending them down in the post, a variety mightn't hurt. Like maybe the hardwood would last a little bit longer in the post, because sometimes it just tends to mush, by the time it gets here.
They also hold up when they're in the propagation house as well. They'd generally they wouldn't want to bend over at the tip, and then you lose it. I just do wonder also, Brandon, with the cuttings you get, and you're on the ground collecting these cuttings, do you always end up with a relatively straight growing tree or shrub?
It sometimes depends on the species, honestly. So Rhodamnia sessiliflora, for example, we've taken bajillions of cuttings of. We have so many of those, and they almost always within a year straighten and correct themselves. Whereas the Lindsay myrtles we've seen the opposite. They almost always spur out everywhere, and I suspect in the future they're not going to form a true taproot. Obviously, cuttings aren't renowned for that, but we are going to have some issues of these larger trees maybe having to be cut back so they don't fall over. But they can still, obviously, a cutting's a mature plant, so a 1 meter, what's typically a 40-meter tree at 1 meter can still produce seed and everything because it's a 40-meter aged tree at the cutting.
Yeah.
So
yes. At least you'll be able to collect seed. So that's the end goal for some of these.
Yeah.
If you can't get a nice straight plant out of it. So
yeah. And that's it for us. It's conservation as opposed to aesthetics. It'd be great to have a boulevard of Stockwella, and it'd be great to have a boulevard of Lindsay myrtles. They have this stunning purple leaf, but at the end of the day, the main goal is to propagate from them and to be able to produce more from them.
I have noticed in Singapore Botanic Gardens, they were also doing lots of rainforest trees, and they were always using hardwood cuttings and serious amounts of fog and water and everything else, to the point where I thought it was too wet because the poor plant is drowning, and they did suffer heavy losses with the hardwoods, but they got lots of plants out of it in the end. I think it's a numbers game. And
we also find hardwoods produce a much larger plant, much faster. They've got a lot more reserved energy, so they're starting quite a way ahead in the race. So you're getting these sort of 3 to 4 meter trees from a cutting that you would otherwise take twice as long to get. So that's been another big advantage.
Yeah, that's good.
Thanks.
Veronica?
Hi, Brandon. Thank you. That was incredibly thorough and really interesting. I'm curious about what kind of interest you've had from other universities in what you're doing.
So we have a group called the IRU. I'm going to pretend I know what that is, but I don't, even though I'm a member of it. It stands for something about sustainability. It's 7 universities that have teamed up to pretty much look at how we can partner around sustainability. And I think La Trobe's on it, University of Western Sydney's on there, Griffith's on there. And I've been asked to give a similar presentation to this around sort of the larger ex situ phase, not just myrtles, for them to replicate. But we're also working with other universities around other programs. So the PsyCab program we're working on, for example, is actually being led by a staff member of University Western Sydney. So we're doing the conservation cultivation side, whereas he's doing the cataloguing management side, and we answer to him in particular. So there is a lot of open collaboration for different programs, conservation programs, and we've certainly shared information with other universities. And I know La Trobe's already got a fantastic nursery set up and a great restoration program in place. And I believe Western Sydney actually has a quite large agronomy centre as well, and that they're doing a little bit around restorative ecology as well already, just outside of threatened species, more so around generic ecological restoration practices. So certainly other universities are working in this field. It's just we've kind of locked into threatened and at risk, whereas a lot of the other universities took a broader range of entire ecosystems.
Fantastic. Thank you.
I've got a question for Brandon. How much space do you have up there in JCU to plant out these rainforest species? Because the biggest challenge of an established botanic garden is space.
We're lucky. We're not established. We've been around since the 70s. We have 386 hectares in Townsville. We have 86 hectares in Cairns. We have 44 hectares in the Daintree. So we've got a lot of space in multiple ecosystem types. And in Townsville, of that, about 100 hectares is landscaping. And luckily for me, a lot of our landscapes were quite generic. So it was sort of like just the same Harpoolia pendula absolutely everywhere. So we've been, and Lomandra, so much Lomandra. So we've been actually thinning existing gardens in a lot of these cases and removing these sort of very prolifically used trees to replace them with these species, which we've been able to because they were just generic, the same species. And there certainly wasn't a strong focus around botany in our landscaping historically. So we've got a lot of really large established spaces that our director has been fully supportive of. Yep, swap that for that as you see fit, have at it. But we've also got in our master plan, that 50-year plan, another 60 or so acres of landscaping and gardens going in over the next sort of 20 years. So we've certainly got a lot of larger spaces to be considering about how we want to use these things. And we've got a boulevard that runs down the whole centre campus for, I think, 800 meters that's on the list as well, to be put in with understories as well, and trees and the whole mix. So certainly
That's
a lot
amazing.
to play with.
It is, yeah. I wish we had more land to play with, but anyway, that's the way it is. Thanks.
Yep.
Any other questions for Brandan?
I think that's it.
I've just got one question as well, Brendan, if you don't mind. I'm not too quick with the hands up or whatever, but more just a question, Matt from Melbourne Gardens, Botanic Gardens as well, and we deal a lot with Myrtle Rust here as well, and thanks for the talk. It's always very interesting to see how you're responding to it. You highlighted that you're a fairly dry environment and an almost void of Myrtle Rust, which is a very fortunate position to be in. But I guess you're doing almost the contrary to what we're doing here, which is actually identifying where our species are and trying to move them away from pathways, particularly, I guess, in then having to respond with a spray regime. So I'm just sort of interested, you've highlighted that you're going to move some of your highly susceptible and key sort of identifying species for Myrtle Rust close to pathways so you can spot it. How do you then respond to it, like with treatment, I guess, post identification?
So we do have fungicide. We've had a handful of outbreaks on those actual Eugenia, and that's it. And we have treated those. We're talking about 1 or 2 cases a year, and I'm pretty sure it's the same 2 plants from memory anyway. Funnily enough, near our science and conservation building. Coming back from all the field work, I'm assuming, our staff. So that's how we've dealt with that. But Cairns is a very different story. Cairns is rainforest. And so our Cairns campus, we did actually go through and selectively remove hundreds of Eugenia from gardens because it was just a smorgasbord of rust. So we said, right, we know that these Eugenia are cultivars from various different nurseries. They're not wild form. They have no conservation value. We actually don't even think they're the Australian form. We're pretty sure they come from Fiji or somewhere, looking at the form. So we just said, right, they're not worth as much as our Syzygium and our Rhodam rhodomotus. So we selectively culled the low value myrtles in that collection we kept a few, but we culled the majority of them to prioritize the species that we really cared for. And we've also got a natural population of the endangered Rhodamnia sessiliflora on the Cairns campus. So we've been collecting cuttings from individuals up there to bring back to Townsville because they weren't fruiting, to then have mass fruiting. And we've now grown on about 600 individuals from those cuttings we took from our individuals to be put back into the wild as seedlings. And we're going to be monitoring how they go. But we're going to be spacing them out and putting other species of non-mutaceous lineage between them as well, and seeing how that works. But yeah, for the Cairns campus, it's a very different approach to Townsville. They're opposites.
Good to know. Yeah, it sort of seems similar to, I guess, the way that we're sort of approaching ours, looking at our collections more holistically, known provenance. And unfortunately, a lot of our historic records were all destroyed. So, there's a lot of really interesting, amazing plants, could have been well collected from other parts of the world. Lophomertus being a primary one that is a key species that we get a lot of Myrtle Rust on here, not Australian, but significant and still threatened within New Zealand as well. So yeah, it just seems that you're handling it in a similar way. So thanks for the chat.
Yep.
Lloyd, did you want to ask another question?
Yeah, I just wanted to quickly follow on from what Matt was saying as well, and just we had a very productive meeting with Veronica. Hello, Veronica. Me and my colleague Bronte, and that kind of helped form our strategy a little bit about finding out what we've got and what's valuable and what's not. But I just wanted to come back to a point that Brandan made about research. And one of the things that came out of our very productive chat with Veronica was, we should try and have a look and get some genetic testing done, because we have those facilities here, which is very handy to have a herbarium a couple 100 meters away. And it's just a funny side note that when we went to go and genetic sequence them, they go, "Oh, we've already done it," because there was someone doing research on hyperaccumulation of Gaucher acminoides, I think, and then for a reference something in the same genus. I'm not exactly across how that genetic testing side of things goes, but they need something from the same family, and they just happened to use a Rhodamnia rubescens in 2018. So now we have genetic records attached to that as well. But it's awesome, and it's, as you say, it's a lot cheaper and significantly less exciting than going in the field, but can be very useful sometimes.
It's also actually certainly an issue that we had here. I think all larger organizations have this, where we have sort of gone out, spent a considerable amount of money covering a project, only to get halfway through it and have someone say, "I already did that. Here's all my data." So I think all organizations have that issue when they get too large. So we're trying to do a centralized database now, where we've got our academia, our research, our adjuncts inclusive even, where they're restricted as to what they can see. So not everyone at the university, only myself, my officer, and one other person can see the lineage of everything we have. But they can see everything we have. And then if they're working with, say, orchids or something, they'll just put a request through our internal system or the external group, same thing, saying, I'm working with orchids or I'm working with these threatened species and I need to know the location of them, and we'll verify the legitimacy of it and then share that data with them and give them access to it. But we've certainly made a centralized system that all divisions within the university now use, not just our division, which I think was a big issue that we have. I think a lot of other groups have these sort of multifaceted divisions that have their own systems that don't talk to each other.
That's great. Anybody else holding back on a question there?
No, I don't.
I think we'll say a big thank you, Brandan, and we'll get this up on the web and make it available to people. And thank you for those of you who could make it today. We had quite a lot of registration, so I'm hoping we'll see those people come through the YouTube channel to sort of get that sense of how it's all worked, Brandan, and some of those really useful lessons and challenges. So thank you very much.
Thanks for having me.
Thanks, Brandan. Thanks, Pip.
Thanks all.